Hi, and welcome to my channel and to this series exploring landmark experiments in protein science based on my book of the same name, which can be purchased on Amazon, and that link is in the description. And this lesson will focus on the discovery of cells and the beginnings of genetics. In the 17th century, there were no tools to study biology, and no one could imagine that anything existed that was smaller than what the eye could see, or that the human body was made up of billions of microscopic living entities, all with specific roles and specialized functions. And within these, even smaller molecules that perform all the functions that a cell requires to survive. The first significant step towards the elucidation of the inner workings of cells and to the discovery of cells themselves was the invention of the microscope in the early 1620s. So at the time, Robert Hooke was an assistant to the famed chemist Robert Boyle at Oxford University, and he thought he could alter Galileo's telescope to look at small things instead of things that were far away. So in the preface to his Microscopial, published in 1665, Hooke expressed a desire for the science of nature to step away from being a work of the brain and the fancy, and return to the plainness and soundness of observations on material and obvious things with the help of instruments that improve the sense. In addition, he shared his excitement in the discoveries now possible with the help of new technologies such as the microscope. The next care to be taken in respect to the senses is the supplying of the infirmities with instruments and, as it were, the adding of artificial organs to the natural. This in one of them has been of late years accomplished with prodigious benefit to all sorts of useful knowledge by the invention of optical glasses. By the means of telescopes, there is nothing so far distant but may be represented to our view, and by the help of microscopes, there is nothing so small as to escape our inquiry. Hence, there is a new visible world discovered to the understanding. By this means, the heavens are opened and a vast number of new stars and new motions and new productions appear in them, to which all the ancient astronomers were utterly strangers. By this, the earth itself, which lies so near us under our feet, shows quite a new thing to us, and in every little particle of its matter, we now behold almost as great a variety of creatures as we were able before to reckon up in the whole universe itself. To improve the brightness of the microscope, he used a convex glass, one whose sides is made rough by being rubbed on a flat tool with very fine sand, through which light could be efficiently concentrated in the very small opening of the aperture, which very much augment a convenient light. And after trying a variety of different configurations, he found that the type of microscope which yielded the best results was 6 or 7 inches long and contained 2 lenses encased in a brass tube filled with very clear water. The specimen was placed here, and Hooke concentrated the light from a candle by passing it through a globe filled with water. One of the samples he looked at in Microscopial was a thin cross-section of a clear cork which was cut using a penknife sharpened as keen as a razor, leaving the surface exceedingly smooth. When he placed these cross-sections on a dark plate, he observed an array of pores much like a honeycomb separated by interstitia or walls. When he cut another section in the opposite direction so as to reveal the length of each pore, he found that each pore was of limited length and resembled little boxes or cells. He determined that there were about 60 of these cells aligned end to end in 1/8 of an inch of cork, and therefore 1 cubic inch should contain over 1,200 millions, a thing almost incredible did not our microscope assure us of it by ocular demonstration. Hooke correctly reasoned that cork was light due to all the cavities being filled with air, which also seemed to explain why solutions could not traverse through it, since this air was perfectly enclosed in little boxes or cells distinct from one another. However, he admitted that we must grope, as it were, in the dark and only guess at the true reasons of things. In 1839, Theodor Schwann published a monograph in which he compared the development of different plant and animal cells into different tissues with an aim to prove the accordance of the elementary parts of animals with the cells of plants. He also acknowledged that his conclusions were largely based on the research of his contemporary and friend Matthias Schleiden. And from this monograph emerged the central principle of biology, which is known as the cell theory and generally attributed to Schwann and Schleiden, that the cell is the most basic unit of life, all living things are composed of one or more cells, and all cells arise from one individual cell. Descriptions of its structure inside the cells came as early as 1682, but it wasn't seen as a separate entity, neither in form or in function, from the rest of the cell, and it wasn't until 1833 that Robert Brown, the custodian of the botanical collections at the British Museum and later the president of the Linnean Society, gave the structure a name, calling it areola or nucleus of the cell, as perhaps it might be termed. He was also the first to formally define it as a separate structure. In each cell of the epidermis, a single circular areola, generally somewhat more opaque than the membrane of the cell, is observable. This areola, which is more or less distinctly granular, is slightly convex, and although it seems to be on the surface, is in reality covered by the outer lamina of the cell. There is no regularity as to its place in the cell. It is not unfrequently, however, central or nearly so. And in 1838, Schleiden suggested that the nucleus of a cell was a universal elementary organ in vegetables, and hypothesized that it must hold an important place in the development of the cell. But the first significant contribution as far as identifying the contents of the nucleus came from Walter Fleming in 1879 while studying salamander larvae. He tested a variety of reagents to fix cells of fin and gill skin, which is a process that halts all biochemical reactions and preserves them in the structural state at the time of fixation, settling on picric acid. He then stained them with a hematoxylin dye solution, which readily labeled the granular structure within the nucleus, and because of this, he named it chromatin for the Greek chroma, or color. With this technique, Fleming identified distinct phases of the nucleus during the life cycle of a cell. The appearance of a fine basketwork of tightly wound threads characterized by a coherent, dense, and regular framework of delicately spiraled threads. A loose coil or basket form of the mother nucleus in which the visible granules gradually become thicker and more isolated, and soon one clearly recognized coiled threads, which were noted to be connected in pairs. These are now known as prophase. An astral form of the mother nucleus in which a typical stratification of the threads is apparent, that is to say, there occurs central and peripheral bendings of the threads. Subsequently, the threads were observed to divide themselves in half lengthwise, lying almost exactly parallel. An equatorial plate in which a grouping of the threads occur in such a way that all the elements, at first somewhat coiled, but later stretched out more and more parallel to the division axis, fills up the space of a thick plate comprising about 1/5 of the entire cell length. The plate always lies in the equator and is oriented at right angles to the division axis, and these represent what we now know as metaphase. A separation of the nuclear figure in which the two nuclei move apart, the actual separation having already taken place in the previous stage. A star form of the daughter nuclei, in which the threads of the two nuclear halves move further and further apart so that some of those situated in the periphery often attain an orientation towards the poles of the cell. In addition, he noted that the constriction furrow frequently makes its appearance in one side of the cell body, and this is now known as a mix of anaphase and telophase. A wreath and coil form of the daughter cell in which the daughter cells form, and a reticular form of the daughter nuclei reversion to the resting condition in which the daughter cells assume a configuration similar to that of the parent cell before division, which is now known as cytokinesis. These threads were subsequently given the name of chromosomes or colored bodies due to their affinity for dyes. One of the major biological issues of the 1800s was defining what a cell was and what it was made up of. But in 1856, Franz Leydig proposed that a cell was a substance primitively approaching a sphere in shape and containing a central body called a kernel, or as we now know it, a nucleus. The cell was thought to consist of two separate components, a central nucleus surrounded by the protoplasm, which was characterized by a translucent, viscid, or slimy material dimly granular under the lower powers, minutely fibrillated under the highest powers of the microscope, which moves by contracting and expanding, and which possesses a highly complex chemical constitution. The first direct evidence that cell membranes were separate structures from the protoplasm came in 1921, when Robert Chambers greatly improved microinjection techniques and injected cells with a variety of solutions. The technique available to him at the time was to inject mercury into cells using a glass pipette, but the pipettes left nothing to be desired, both in terms of their size and ease of making, and the method was very difficult since injection depended on the expansion of the mercury, which was difficult to control. Therefore, he built an instrument that helped stabilize the needle while providing fine control over its position via the adjustment of screws. So on the left you can see a side view of the apparatus, and on the right a top view. The specimen is placed at the end of the needle at D, and screws I, G, and H are used to make adjustments in 3 dimensions. Using this instrument, Chambers injected starfish eggs with neutral red dye, a dye that changes from red to yellow in a basic environment, so a solution with a high pH, and then soaked the cell in a basic solution. And under these conditions, he found that the cells remained red, which suggested that the basic solution could not enter the intact cells. But when the solution was injected directly inside the egg, there was a color change where the dye was injected, which spread throughout the cell but stopped at a film surrounding the protoplasm. This suggested to Chambers that there must be a structure around the protoplasm that was made up of different components from the protoplasm itself. Following Chambers' experiments and inspired by Overton's suggestion that the cell membrane might be composed of lipids, Gregor and Mendel set out to better characterize the cell membrane and its components. Here they use a method developed by Irving Langmuir based on the fact that when oil is placed on top of water, it spreads out and occupies a measurable surface area. So Langmuir placed water in a tray with glass rods inserted through a paraffin-coated strip of paper, which prevented the paper from getting soaked, and across the rods was attached a support with a knife, where a pan containing a weight was hung. The weight caused the glass rod to tilt to the left to a degree determined by the weight in the pan. Oil was then placed on top of the water between the glass rod and another strip of paper, which he started moving towards the glass rods until the lipids were so compact as to push up against the strip of paper, at which point the glass rods started to straighten. The surface area between the strips of paper was then measured with a ruler. Using this technique, Gregor and Mendel calculated the surface area of isolated lipids from red blood cells and compared their value to the theoretical surface area of a cell. The authors found that the lipids occupied twice as much area compared to cell surface, which implied that the cell membrane was composed of 2 layers of lipids, a lipid bilayer as we now refer to it. Given these results, they suggested that the lipids, which are composed of one hydrophobic end, which wants to stay away from water, and one hydrophilic end, which likes to associate with water, were arranged in such a manner where the hydrophobic tails of the lipids faced one another at the center of the bilayer, whereas the polar heads were directed towards the water. Although the lipid bilayer described by Gregor and Mendel would eventually be shown to be correct, it would not happen until the advent of superior techniques in the 1960s and 70s. In the late 1800s, most of what was known about cell biology came as a result of improvements to the microscope. However, Gregor Mendel, an Augustinian monk now known as the father of modern genetics, went in a different direction and instead was interested in testing the blending theory of genetics, where apparent traits were thought to be diluted and blended together to create offspring with a mixture of traits in a random manner. Farmers had long known the outcome resulting from generations of crossbreeding sought-after traits, so Mendel studied the inheritance of simple P traits, or phenotypes as they're called in genetics, such as color, size, shape, or plant height, by analyzing the results of crossbreeding experiments and determining if a precise law could be deduced to explain how traits were passed down from one generation to the next. His experiments were painstakingly and meticulously performed over 8 years of crossbreeding thousands of peas, and his analysis shed light on the mechanism of genetic inheritance. First, he had to determine which traits could be used for his experiments, using only those that had clearly distinct phenotypes so as to be unambiguously identified. Importantly, he had to protect his plants from pollination from neighboring crops to not ruin his own experiments. So he started by crossbreeding 2 versions of a particular trait, for example, a tall plant with a short plant, to see which phenotype the progeny would take on from one generation of crossbreeding to the next. The first generation of offspring, called the hybrid generation, only expressed one phenotype for each pair of phenotypes tested, so in this case, only tall plants. This already discredited the blending theory, since we would expect all plants to be of medium height according to this theory. When hybrid plants, or the F1 generation, were self-pollinated, the phenotype of the next generation, the F2, followed a recurring ratio. 3/4 of the progeny expressed the phenotypes observed in the F1 generation, or tall plants, whereas 1/4 expressed the phenotype from the parent generation that was missing in the F1 generation, so in this case, the small plant. To explain this, Mendel proposed the term dominant to describe those characters which are transmitted entire or almost unchanged in the hybridization, and therefore in themselves constitute the characters of the hybrid, and recessive for those which become latent in the process. When the F2 plants were self-pollinated, the offspring had phenotypic ratios that depended on the parent. The F2 short plants, 1/4 of all F2 progeny, only produced plants with the recessive trait after successive generations. 1/3 of tall plants only yielded plants with a dominant trait after multiple generations, but surprisingly, self-pollination of 2/3 of the F2 plants with a dominant trait, or half of the F2, yielded an F3 generation in which 3/4 of the plants expressed the dominant trait and 1/4 of the plants expressed the recessive trait, a ratio identical to that found in the F2 generation. These results led to the conclusion that the recessive trait, although seemingly lost in the hybrid generation, was in fact not lost, it was simply not expressed. Although the results of Mendel's experiments were groundbreaking in retrospect, they made little impact following their publication in 1866. It was only years after his death in the early 1900s that Mendel finally received his due credit. Although it was clear by this time that the nucleus was somehow involved in heredity, the extent of its importance and in fact its actual role remained elusive. But in 1903, Walter Sutton proposed a role for chromosomes in heredity, noting that it has long been admitted that we must look to the organization of the germ cells for the ultimate determination of heredity phenomena. Nearly a year ago, it's become apparent to the author that the high degree of organization in the chromosome group of the germ cells could scarcely be without definite significance in inheritance. Many points were discovered which strongly indicate that the position of the bivalent chromosomes, or a matched pair of chromosomes from each parent, in the equatorial plate of the reducing division is purely a matter of chance, that is, that any chromosome pair may lie with maternal or paternal chromatid indifferently toward either pole irrespective of the positions of either pair, and hence that a large number of different combinations of maternal and paternal chromosomes are possible in the mature germ products of an individual. He also proposed that chromosomes were composed of smaller entities that were responsible for defining the characteristics imparted by the chromosomes. And so with this in mind, Sutton calculated that over 68 billion different gene combinations could result during fertilization for an organism with 36 chromosomes. In contrast, there are 46 chromosomes in humans, which means that about 7,000 trillion genetic combinations are possible. In the years to come, it would be shown that indeed, each chromosome is composed of many distinct entities called genes, which are heritable and which determine the phenotype of an organism. This work on heredity and chromosomes led to the formation of the laws of inheritance. Law of segregation. During cell division, each copy of a gene separates so that each daughter cell receives one copy of each gene. The law of independent assortment. Copies of different genes segregate independently from one another, and the law of dominance. The genetic form that a gene can take can be either dominant or recessive. As such, an individual carrying one dominant allele, or a version of that gene, will express that trait, whereas two recessive alleles are required for its trait to be expressed. Thomas Hunt Morgan started using fruit flies in the early 1900s with hopes of inducing mutations in them to further study genetics. So he took on 2 undergraduate students to help set up what became known as the fly room at Columbia University. There, Morgan and his students subjected his flies to numerous conditions and reagents to try and induce mutations: salts, sugars, acids, alkali, etc. And after 1 year of breeding multiple generations of his flies, Morgan found a white-eyed male mutant among the red-eyed flies. Morgan called these red-eyed flies wild-type flies, since they could be obtained in the wild and was therefore of typical genetic background. Surprisingly, breeding this male mutant with a red-eyed female did not result in any white-eyed progeny. However, When the F1 was crossed with itself, the mutation reappeared in about 1/4 of the males but in none of the females, a Mendelian ratio for the phenotype in males. But when he crossed the original mutant fly with an F1 female, 1/4 of both males and females had white eyes. This indicated that females were not excluded from this phenotype, but were somehow less prone to it. Morgan called this phenomenon sex-linked inheritance. Another experiment was performed to further shed light on this phenomenon. He crossed a white-eyed female with a red-eyed male, which led to the most surprising result. All females had red eyes and all males had white eyes. Analyzing the results of these and other experiments, which included similar results for other sex-linked traits, he concluded that this difference exists because one of the factors for the sex-limited characters in question is absent in one of the female's determining chromosomes, while the genes for the secondary sexual characters of the male are contained in other chromosomes, possibly in those that contain the male determinants. Morgan suggested that female flies have 2 X chromosomes and males only 1, such that a female inheriting a recessive factor such as white eyes, and these factors would eventually be shown to be genes, could have a dominant factor on their second X chromosome, resulting in the dominant phenotype, whereas a male having only 1 X chromosome would express the phenotype of whichever factor he received, whether it be wild type or mutant. Morgan also suggested that women have 2 X chromosomes and men only 1, since the fruit fly's wide-eyed inheritance patterns closely resembled that of color blindness in men. We now know that female mammals do have 2 X chromosomes, but then males have a Y chromosome as well as a single X chromosome. And so to summarize today's lesson, cells are microscopic entities made up of a central nucleus, a protoplasm, and a cell membrane composed of a lipid bilayer. The nucleus contains the chromosomes, which are thought to be at the heart of genetic inheritance. A specialized type of cell division called meiosis results in daughter cells called gametes, ova and spermatozoa, having only 1 copy of each chromosome. Gametes in each species carry a distinct number of chromosomes, 22 in humans plus 1 sex chromosome, which combine upon fertilization and results in the new cell having 2 copies of each gene, 1 from the mother and 1 from the father. The expression of a particular trait depends on the inherited alleles or version of the gene. As Mendel determined, in its simplest form, the phenotype of the dominant allele will be expressed unless the organism has 2 recessive alleles of that particular gene. However, unbeknownst to Mendel, a phenotype is usually defined by the alleles of 2 or more genes, and genes that are additive are also common. As mentioned earlier, the fertilized egg carries either 2 X chromosomes, giving rise to a female, or 1 X and 1 Y chromosome, giving rise to a male. This is the case for most mammals, but may be different for other organisms. It should be noted that certain mutations can occur in which an XY individual is physically female and where an XX individual is physically male, and these are expected to be more common than previously thought. Soon after fertilization, each chromosome is duplicated and aligned at the center of the cell, where 1 copy of each chromosome is pulled to either pole of the cell. The egg then undergoes cell division, and each daughter cell receives a full complement of chromosomes. And this brings this video to an end. I hope you enjoyed it, and if you did enjoy it and would like to know more about this, please check out my book using the link in the description. And if you have any questions, please leave them in the comments and I'll do my best to answer them. And I hope you'll join me for the next video on the discovery of proteins and the elucidation of its molecular structure.